We'll conclude this module with a brief video. It may not seem brief as you're going through it, to discuss macromolecular structures. And this is sort of a synthesis of all we've learned about bonding and intermolecular forces, but it allows for some truly remarkable molecular structures that support the existence of life itself. Our learning goals in this case will be the organization of macromolecular structures into hierarchies, and how the basic ingredients of protein structure arise from those hierarchies. So let's look at hierarchies of structure. Okay, first of all, we have a primary structure in these macromolecules that is essentially the covalent bonding that creates a long linear chain. Most of these macromolecules are biopolymers, which means they're a long chain molecule held together by covalent bonds. There is also a secondary structure which arises really from hydrogen bonding interactions between different parts of that long biopolymer. A tertiary interaction then is steric interactions that then shape and mold the greater shape, if you will, of that macromolecule into a distinctive shape that gives it its functionality. And finally, there's a quaternary structure, which is how these various macromolecular structures pack together, sometimes in multiple independent units. So what kinds of biomolecules do this? Well, for example, proteins. All proteins are macromolecules that are built with this hierarchical structure. Also nucleic acids like DNA. Now I should mention by way of analogy that this is not dissimilar to how a city, a large urban center, is organized. There's a primary structure which might be the street with some houses on it, secondary structure which is a neighborhood, which is a gathering of these streets that includes these neighborhoods, a tertiary structure which might be regions of the city, and a quaternary structure which describes how these different regions of the city are related to one another. So you can think of this hierarchy of structure in that way if it's helpful to remember what the various parts mean. So we'll start by talking about the primary structure of proteins. Proteins are amino acids that are connected together to form a long biopolymer. An amino acid is basically a molecule that looks like this. It consists of an amine part, which is the NH2, a carboxylic acid part, which is the COOH, and we're going to label that carbon that's attached to the carbonyl carbon as the alpha carbon. Next to that alpha carbon, or attached to that alpha carbon, is a side chain or residue for that amino acid, and different amino acids will vary depending upon what the identity of that side chain residue is. These residues are connected by amide bonds or peptides. Okay, and this happens when these molecules undergo a condensation reaction where they connect between the nitrogen on one amino acid to the carbonyl carbon on the other, and they release water as a side product. Okay, this is shown below. So it's a dehydration reaction, if you will, and the net result is the formation of a new peptide bond between the carbonyl carbon on one amino acid and the amino part of the other. Now this can happen over and over again because every amino acid has one of each bond, so that's how it creates a long polypeptide chain. I should note that these two carbons here are chirality centers. And so it really is critical that they have a particular handedness associated with them. And because of that handedness, it's going to lead to characteristic handedness of different structures that proteins can form. So what happens when we bring this linking process together many times? So we start with one amino acid, we connect another, and then another, and then another, and so forth. All right, so this can go on for many thousands of residues, in fact. Now we're going to indicate that each residue can be distinguished from each other, but they're distinguished largely by the identity of the side chain, the R group that's attached to that amino acid to begin with. So the net result here, as I mentioned, is a polypeptide chain. And these bonds in here that connect the different residues to one another are peptide bonds, or you can also think of them as amide bonds. All right, when it folds into its active shape, that's when this polypeptide chain actually becomes a protein. It's not really functioning as a protein until it is in its proper shape. Should also note that one end of this chain terminates in an amine group, That's the N-terminus. And one end terminates in the acid, and we call this the C-terminus. All right, so this is some of the language that we use in talking about these polypeptide chains. Now these chains can form secondary structures, and that happens when there is hydrogen bonding between residues on different parts of that same chain. So you can imagine if it coils around until one part comes near another, we can now form a hydrogen bond, say, between these two parts, the amine part of one, the hydrogen from the amine, with the oxygen of the other. Okay, when it coils properly, these hydrogen bonds can form four residues apart in a helical shape. So if we look at the straight chain polypeptide, in other words, these are the oxygens that are going to form hydrogen bonds, and these are the nitrogens that might form hydrogen bonds, or the amine parts that will form hydrogen bonds with another part of that chain. Okay, when all is said and done, and I'm going over this rather quickly, but when all of this is said and done, it ends up creating this helical shape. Now the helix has been emphasized here by drawing the ribbon through it, but you can see the underlying molecular structure to it. These are called alpha-helices, and the structure is common to a huge number of proteins, virtually all that I've looked at in the protein database. The coiling geometry is very consistent from one protein to another. It is always right-handed, and this is due to the chirality of those chirality centers on the amino acids. There are also 3.5 residues along each turn of this helix. So you go from one coil to the next, you're going to go through 3.5 of these amino acid residues. The vertical distance between these turns is always around 540 picometers. So it's that distance there. The residue groups are going to be external to the helix, so you can see them sitting out here on the outside. And that's important because they're the things that really determine the chemical functionality of a protein. Because they're sitting on the outside, they're the ones that are interacting chemically with other parts. The other piece, the residue chain going through there, the polypeptide part, is really just a structure to hold it together. But it's the residues that, for example, cause it to fold into a particular shape and also give it this chemical reactivity. Now there's a second type of secondary structure, and it's called beta sheets, and these occur when strands of the polypeptide chain align parallel to one another. So here's a picture of that. These are two parts of the polypeptide chain, and they're aligning parallel, and in this case we can see that we have hydrogen bonds that are forming through several adjacent parts of the polypeptide chain. So this forms a pretty good connection and leads to some stability. It holds these parts of the protein's chain together. Now we usually designate these things by pointing an arrow from the N-terminus to the C-terminus, so we think of a handedness, if you will, or a directionality to these polypeptide chains that goes in these directions. So you'll notice that these two are oriented in opposite directions, so this is called an anti-parallel strand. It is also possible to create beta sheets from parallel strands, although the structure is slightly different there. But I want you at least to be aware of these beta sheets, and I'll try to point them out in some pictures that we'll see presently of protein structures. Now there's also a higher order structure in proteins, and that's what we'll call a tertiary structure, and it's what happens when protein folds on a larger scale. And the shape of the protein dictates its functionality. So now here is a picture of rhodopsin, which is the protein that's in your eye and helps you with vision. And you can see the retinal parts that are attached to the rhodopsin there are detailed as little molecular diagrams attached to the ribbons and coils. The coils on here are all alpha-helices, and they're colored purple in this case, and then the blue parts are indicating the presence and the location of beta sheets in this protein. So you can get a feel for how to view one of these protein diagrams. What you're looking at really is the secondary structure of the protein as expressed through these coils and these broad strands that represent the beta sheets. Now proteins serve as enzymes that are specific to a particular substrate. That is to say, they are going to direct a particular kind of chemical reaction. And many times they have a metal cofactor embedded in them, and those are called metalloproteins, and we'll talk about that more in Module 8. But here's an example of carboxypeptidase, which has a zinc embedded in it. The zinc is colored purple in this particular picture, and it's at the active site of this protein. So that's where the interesting chemistry is going to be facilitated by carboxypeptidase. Finally, we have proteins in a quaternary structure, and this involves packing of independent proteins and polypeptide strands together to form some sort of greater functionality. And some of these structures include groupings of multiple strands. So for example, we have hemoglobin. Here's a picture of hemoglobin, and each of the different polypeptide structures is colored differently. So you can see there are 4 of them, one that's green, one that's yellow, one that's blue, and one that's red. They're all actually chemically identical to one another, and they are the globin part of this hemoglobin protein. The heme part are detailed as little molecular parts that are hanging out there. We'll talk about this more when we get to module 8, but this is the general structure of the stuff in your blood that helps carry oxygen to your cells. So finally, let's review the hierarchies in proteins. We have a primary structure that is the polypeptide chain with amide bond linkages. There's a secondary structure caused by the hydrogen bonding between different parts of that strand. This gives rise to structures like alpha-helices and beta sheets. There's a tertiary structure which is really formed by the steric interactions between different parts of that complex protein. So it's different pieces swinging away from each other so as to give each other space so that they're not overlapping. This is actually what creates the active geometry for the protein and causes it to be useful for us in terms of facilitating reactions. Finally, there's a quaternary structure which involves the packing together of these multiple proteins or polypeptide strands. And this is what creates sort of a supramolecular structure, and hemoglobin is a great example of this. So I hope this has given you some feel for the macromolecular structures and how proteins are a great example that exemplifies all of these various pieces of the hierarchy.